research seminar of the Center of Excellence in Biodiversity and Natural Resources Management. On behalf of the director of the center, I welcome everyone here present. And today we are glad to be with Dr. Egide from the Department of Biology, School of Science, College of Science and Technology, and the research associated to the Center of Excellence in Biodiversity and Natural Resource Management. And Egide is going to do the presentation on the variability of airborne microbial communities and the correlation of chemical air pollutants and atmospheric particulate matter in Rwanda. Dear Dr. Egide, we are very happy to be with you, and I welcome you for the presentation. For those who are present in the seminar, you can post your question if there is in the chat room, or you can write it somewhere and ask the question after the presentation.
The air pollution remains the largest environmental cause of premature human mortality, even larger than AIDS and malaria combined. So my question, I was wondering why we set all the measures, but the air pollution keep increasing. Couldn't be some other maybe association with other pollutants in atmosphere that could actually contribute a little bit to the effect of this pollutant. And this is where I started this research, tried to look the correlation between chemical and biological composition of air pollutant. Yeah, it's like give me a sample of the air and then I do chemical and biological analysis and tell you what is there in air. We care about air pollution because, as I said, 7 million die every year and 4.2 million died from ambient or outdoor air pollution, while 3.8 also die from household air pollution. Air pollution not only affects humans, but also affects climate change. They cause acidic rain and so on. We have quite a lot of pollutants, but there is certain pollutants which are the pollutant of the greatest health concern. And here we have nitrogen dioxide, ozone, sulfur dioxide, as well as particulate matter. As you see, this particulate matter highlighted alone, this is actually the most pollutant of the greatest concern worldwide. Even evaluation of air pollution in a country is only measured by monitoring this particulate matter. This particulate matter, it's a major concern because it can affect the climate, urban climate, and cause what we call urban heat island. It affects meteorology, cloud formation, precipitation process. It affects air quality, visibility, affects health impacts, respiratory diseases, cardiovascular diseases, asthma, lung cancer, and so on. It can also cause the acid deposition that destroy crops and the destruction of cultural monuments. As well, it affects industrial activity. So it almost affects everything. That's why regardless of thousands of pollutants available, the particulate matter is the one of the greatest health concern. So now we have only 3 ways we can get the air in our body. Is actually through inhalation of this pollutant or through dermal contact, as well as through ingestion. When you just eat fruit without washing, has been sprayed with different chemicals. Children or teenagers are the most affected because they have developing lungs. They spend more time outdoor, as well as their size or their height is actually, which is really close to the ground. So emission coming from vehicles immediately affects children, as well they breathe more air compared to the adults. The same as aging people, because they don't have that capacity to, their cells are really weak so that they can combat to any pollutant coming their nose, as well those already suffering from existing chronic respiratory diseases, they are really mostly affected. Particulate matter is an issue. So where you see in the red is where we have a large number of the death of people died from air pollution, especially from particulate matter. And you can see Africa along our East Asia are the mostly affected. The dot you see in the map is where we have the monitoring station. Even though our region is more affected, we don't even have the monitors so that we can monitor, see what is the air quality status, and then we can't monitor what we don't measure. So there is still also a problem to see what is in the air. So this particulate is really small. They divided into two sizes. The PM2.5 being small, it can penetrate all the way through the lungs, and the PM10 through the pharynx. So the smaller, the more penetration capacity. So, and in terms of comparison, this is a particulate at really small, 7 times smaller than the size of human hair. So the world recognized this pollutant as the greatest self-concern, and they set different standards to comply by countries. So now, before coming up through how it reacts with biological pollutant of airborne microorganisms, I have to know what are the dynamics of this pollutant, this aerosol in atmosphere. First of all, we have this primary emission. Those are emissions that emitted immediately from source. So if the car emits smoke, it will go immediately to the atmosphere. That's primary emission. So other, they may form through coagulation. The others where they through the condensation or evaporation, as well as this, they can move in atmosphere through dry or wet deposition. But the most important, these small particulates, they are really small. They don't even require a visa to travel from one country to the other, or from one area to the other. So they can travel from long range distance for several miles from one region to the other. So having these aerosol dynamics, and this give an idea that how the airborne microorganisms, which as well also are in terms of smaller size as particulate matter. So once they're in atmosphere, they can coagulate with chemical pollutant, they can condensate, something like that. So talking to the long range transport, I share very recent research I and my colleague were conducting during Nyiragongo eruption, which is long range transport. We're trying to look what are the emissions in Rubavu City, which have been actually emitted from Nyiragongo. So we tried to set this mobile monitoring around the Nyiragongo area. However, during this modeling, we found the emission or the smokes coming from Nyiragongo, it's long range transported away from Rubavu. And that's why we conclude there was no imminent pollutant across the city. However, when we were moving actually across the Nyiragongo area in Rubavu, the emission was a little bit high. But I want actually this to highlight even how this emission are long range transported. So if the wind direction and the wind speed were moving toward Rwanda, so Rubavu could have experienced a lot of pollutant emitted from volcanic eruption. However, the air masses were moving across Goma instead of Rubavu, and that's why there was no less contribution, there's no more contribution of Nyiragongo to the Rubavu city. So this pollutant, particulate matter, is when it is found in atmosphere, it's not only a single pollutant. It contains a complex mixture of air pollutant, and this includes a polycyclic aromatic hydrocarbon. It contains airborne microorganisms, including pathogenic viruses, fungi, and bacteria, as the other also toxic heavy metals. So, however, when I was doing the research, I focused on bacteria, fungi, and this PAH and NPAH. PAH, which stand for polycyclic aromatic hydrocarbon and their nitroderivatives. I focus on this because they are found carcinogenic. So they are really toxic and then can cause the cancer to the humans. So for bacteria and fungi, as well as the contents of different pathogenic species, which also affect not only human, plants, and animals. However, due to the research requirement, I couldn't analyze the viruses. However, because it was really difficult to detect the viruses with the infrastructure I have at the moment. But my research focused on fungi and bacteria in terms of microorganisms, and as well as these toxic polycyclic aromatic and their neutral derivatives. Before doing further analysis, I corrected actually, let's say, the air in the city of Auckland, in New Zealand, in the urban area, in the city center. I correct also another in a typical rural location, 100 kilometers from the city center of Auckland in New Zealand. And then I did what we call the pea staining techniques. And then I found there was a quite coagulation. If you look in the first photo, you can see different blacks dots as well as the whites. So those blacks, they are black carbon, which are emitted from vehicle emissions. And the white is from actually biological composition. However, if you look in the rural area, there was no black carbon. And this is because this area in a rural location was 100 far from the city center. It's an actually only vegetation, no any influence of vehicle emission, no influence from other industrial emission. And that's why you find in the first pea staining on microscope, they can see different black dots which come up from the chemicals as well as other from biological. And this is where I found, oh, these things they can both found in atmosphere. So if I want to highlight this, you can see emissions from actually biological composition, and that's our fungi, which can also be correlated with the black carbon emitted from the vehicle. And then I had a concern that inhalation of this air, which contain the black carbon as well as the pathogenic fungi or bacteria, actually could maybe cause more effect than was supposed to cause if there was no this combination, this correlation between the two. Moving from microscopy research, I came in Rwanda and then tried to correct air quality in different land use. So I went to actually here in downtown where there is the Ministry of Environment on the rooftop. I corrected the air as urban roadside. In a CST, the rooftop of Muhabura building, I corrected as well this air pollutant, particulate matter. And I went in a Musanze in a typical location to collect air in a rural location so that I can see this different variation in terms of chemical and their biological composition. I'm really excited to see what's the variation in different land use. And I used a higher volume air sampler, which correct around 1,000 liter of air per minute. So to make sure air corrected in the urban side in, let's say at CST is aerosolized and it can cover the whole urban background, same as other sites. So this study was quite important because this was actually the first study in Africa, tried to look biological and chemical composition in the air, as well as in Rwanda. So, and this chemical and biological also is really a risk to human. And I suspect that inhalation of the air which contain both chemical and biological composition could cause the more effect that was supposed to do. Now, I spent almost 1 year trying to design methodology because there was no method available. I tried to detect this pollutant, but the biological and chemical together. So I tried to do a collocation of different samples. I tried to set all the measures throughout the whole year until I get one significant method which were able to give the correlation or to analyze both chemical and biological together. So how I did this, my experimental design, I corrected the air using this high volume air sampler. And this air is corrected through the paper filter, a glass fiber filter. And then after correcting from the site, from Musanze, Ministry of Environment and the CST, I have to correct the air, transport it in the lab, store in the fridge before further analysis. So, and then I take that filter, I cut into two parts. One part I analyze for biological and another part for chemical composition to make sure the air which has been captured on the filter is fully on that filter, so that by randomly cutting in half, one half can analyze for chemical, another for biological. But before that, I have done a role of pre-analysis, trying to see does all air is actually saturated on the filter equally, so that to make sure if my half part contain actually the mixture of everything. So before I done a lot of analysis to see that the air is randomly distributed on the filter, and then in any part you can cut represent actually the whole air. So, and then one part I do a DNA analysis from the air, which was quite hard. And then I analyze from biological composition, which is bacteria and fungi. And then I do extraction and I analyze for pH and NPH as a chemical analysis. I do a lot of statistics and bioinformatics to try to see the possible now correlation between the results obtained from both biological and chemical, and then I interpret the data in different way. So for analysis of microbial component, which I analyze for bacteria and fungi, so after the DNA construction, I have to quantify the DNA, and after do PCR and sequencing, and then analyze through using next generation sequencing, a high throughput DNA sequencing. So I didn't do culture technique, I do culture independent, which analyze immediately from the filter and get the data using this MiSeq sequencing, and then do biological interpretation. So before sharing the results from biological, so this is the result from the chemical analyzed in the air. So I found that the mean of this particulate, PM2.5 and PM10, so we're quite higher in the dry season compared to the wet season. And this is actually makes sense because during the dry season, there's quite a lot of dust moving along. So if you look these three panels, the first panel is in the rural in Musanze, the second is in the urban background at the CST, and the last panel down is actually the result obtained from the Ministry of Environment headquarters, the roadside. So you can see we have a lot of concentration, highest concentration at the roadside compared to the urban background, compared to the rural location. So, and then the dry present actually the high concentration compared to the wet. Moving to that, we have to see different source analysis where I perform different traces, different principal component analysis to confirm really the major source of this pollutant, chemical pollutant, and mostly wood burning was the main contributor for air pollution in the rural area, where diesel and gasoline powered vehicles in the urban roadsides, as well as in the urban background. And of course, I have also to perform back trajectory analysis, where on certain time I found there is a day I can find the highest concentration, where there was another, I found the lowest concentration. So what I do a back trajectory high split model, try to see where the air is coming from. And then we found, because of our geographical location of the rural site, we found can capture regional air mass from other East African. So that's why I was saying the air or air pollution doesn't require a visa, can travel from one area to the other. So in that case, I found the highest concentration found in the rural area has also some contribution of air which were long range transported from Southern African savannas fire in East Africa. And of course, we do this research because of human health. As I'm analyzing the pollutants which are able to cause the cancer, I have to do carcinogenic risk assessment, which is the paper published in Environment Science Technology. We found that the WHO, this is the standard, the Gini line is the standard of carcinogenic risk. And we find even the rural location in the urban background and the urban roadside, the concentration or the carcinogenic risk is really high compared to the WHO standard. And this is actually an issue of people living at the roadside. So living people at the roadside was likely to have a definite risk, especially from this emission because of the high concentration of this toxic emitted from the vehicle emission. And this is for people actually who are living next to the road. So bringing the conclusion one of the chemical before I move to the biological analysis, Rwanda has a pollution problem, if you can see, and it's still lack of the data to cover it. However, different measures which I discussed before now it look like it's improving. This was the first empirical air quality evaluation in the region trying to look at this pollutant of the greatest health concern, although it still exceeds the WHO. Of course, we found this excellent correlation between air quality and human activity. This I didn't discuss on Car-Free Day, which I analyzed in that paper, which reduced the air pollution around 30%. And vehicle emission and wood burning are the source of these toxic or carcinogenic pollutant in urban areas as well as in the rural site, and the carcinogenic risks highly exceeded the WHO standard, which come up to the conclusion that immediate emission control is required. Now, as the Rwandan population is increasing, we can see especially the traffic, which is a major contributor of most of these pollutants, where the traffic is heading. And that's why we need also to start looking what are the possible solutions. So come up with biological composition. We try to analyze this biological component. And this is, I use this principal coordination analysis. The red is actually the rural area. The green, it's urban background. And then the blue one is in the urban background. Urban roadside, sorry. So what are the results? So if you do this, it's grouping all the data, biological data together, and then you found they were all separated each part. There was even a very clear separation in terms of bacteria. So the first A and B, that's a 16S, which symbolized bacteria, and the down ITS is a fungi. So on the bacteria, there's a very clear analysis. Even the statistical analysis shows different between this site, rural, urban background, as well as the urban roadside. So what does this tell us? So airborne bacteria and fungi community, which were estimated using this next generation sequencing, it shows that microbial analysis of the species richness and the community structure of airborne bacteria and the fungi community, they vary with site, which is a very clear differentiation between land use, rural, urban background, and urban roadside, as well as through the size, PM2 and PM2.5, as well as we are going to see how this variation also vary between the season. And this is clear because we are living in different landscape hues. You can see that people having living in different environmental characteristics, and as well the contribution of pollution is totally different because of where you live or where we live. Analyzing the seasonal variation, dry and wet, there's a very also significant correlation between actually separation between this species richness and species community. However, it's not clear as we saw for spatial variation, but seasonal variation, although it was significant, but there was quite a difference between dry and wet season, which were not observed actually for fungi. We have to do the analysis of actually the microbial community composition. So the area we detect in the rural, in the urban background, in urban roadside was actually dominated by different phyla, where acidobacteria, as a chlorophyx, firmicutes, proteobacteria were the main contributor of this, in this land use site. Same as the fungi, we have as well different classes where Dodrodomycetes and Agaricomycetes was actually the main also as well contributor in different land use. And then if you go to detail, you can see how we have some dominated the rural area, while others dominated the urban area. So we're able to identify different pathogenic and algic bacteria and the fungi. Some are plant pathogenic, and are animal as well as human pathogenic. And this is clear here. Let's give an example in a red Acetobacteria, which as well is a genus which contains a lot of pathogenic species, same as Bacillus. You can find some were dominated in the rural area, while also dominated in the urban area. So depending on the composition on the source, we can have different species according to the land use. I tried to do a correlation where I found this pollutant, PM2.5, PM10, increase as well with the increasing of the chemical composition, but there was no correlation between species richness and species diversity. But the most important we found that these bacteria, which are well known in atmosphere to degrade the chemical pollutant. In the last table, you may find Acinetobacteria, Methylobacterium, Micrococcus, Pseudomonas. Those bacteria are all found sometimes emitted in atmosphere and has been found to degrade these chemical species. And then this was actually evidenced in this research because all of these bacteria, they have a so-called negative correlation with chemical pollutant. So we try to do what we call redundancy and variation analysis, and then to try to analyze the composition, the correlation between these, chemical and biological. So what did we find? We found most of the microorganisms detected in this study, which has been some associated with long-range transport of dust and plant. However, both bacteria, community structure, and environmental factor explain around 45% of variation of the chemical composition. So relative humidity and the temperature, as well as microorganisms, they explain the variation of the chemical composition or the chemical pollutant around 45%, and the 55% remaining unstudied. I didn't find actually the 55%, what are the major other contributor, which actually call further research to look at the 55% that contribute to the chemical variation, which were not found in this study. So let's bring to the conclusion for biological one. This was the first study to demonstrate the spatial temporal variation of bacteria and the fungal community in the particulate of the greatest health concern using next generation sequencing. So I demonstrate this relationship between microbial community and the structure, microbial community structure and the chemical composition. And this is the first correlation study in the region. And this result showed that the diversity and the composition of airborne bacteria and the fungal community, they may vary with the site, because we found there was different from the rural urban background as well as to the website. It may vary through the PM size fraction. So some were detected in a PM 2.5 and other detected in a PM 10, as well as may vary during the season. So some may vary during the dry, other during the wet season. The result indicated that the particulate matter air pollution level do not affect species richness and community diversity. However, the correlation between the community, microbial community, and this chemical were observed. Around 45 of the total variation of the chemical composition were explained by environmental factors, which is relative humidity, temperature, and the bacteria community structure as well. However, further study is still required to investigate the role of the chemical and biological composition of this PM. Now, as we speak now, Rwandan population are increasing, the size population, especially living in the city. You can tell through this map I'm presenting from around the 80s to 2010 to 2020, how the city of Kigali is expanding. So expanding or increase in this population is associated with the highest increase of human activity, which means the air pollutant is not likely to decrease, but we need stringent measures because of this increase in human population. Some requires international collaboration to monitor this air pollution in Africa. As I said, this analysis of, especially for this biological composition, is ridiculously expensive. And then even one sample can cost several dollars. So, and it requires actually a high-tech technology to be able to detect these microorganisms in the air, where we use this high next generation sequencing, MiSeq, which I don't think we have currently in Rwanda. So as far as they require a lot of expertise in chemical, biological, in physics, so that the people can work together to do this research across the world. And of course, technology, which should minimize these air pollutants. And of course, we have to promote the active transportation, try to use these, reduce this emission, especially in the city center. So if I present some of the solutions which have already have the significant impact, the Rwanda initiated this car-free day, and you can see when I performed this research, we found for a single car-free day actually could save around 10 lives per car-free day. And you can save actually a lot of money which has been spent at the hospital of people suffering from respiratory diseases. So one of the policies actually successful, we can see compared to the Sunday with other Sunday where there is a car-free day, the air pollution could reduce more than 20%. And then if you look during COVID-19 lockdown, I tried to use this natural experience, try to look how the air quality looked like when there was no human activity. And they may really found that during lockdown, where around 80% of human activity were reduced, the air pollution also significantly decreased during the full lockdown. However, nowadays, where there is no measures, you can see how the pollution has dramatically increased, which this gave a clear picture how a human activity are clear linked to the environment. However, despite the reduction of human activity during COVID-19, this particulate remain above the World Health Organization, which I'm not sure if we get another which can, where there is the human activity can reduce up to 8%. And this is called a very strong measure to start to see how this particulate could be reduced. Speaking to that, as well as the viruses also can be found in atmosphere, and also the other research, which I didn't work on the viruses in atmosphere, I suspect as well they may react with these pollutants. So, and then their effect could be actually dangerous. So very recently in a research in China, they found even people which were in most polluted cities where the most have recorded a lot of death during COVID-19. And because of this small particulate, I suspect that once they are emitted in atmosphere, can react with also this small size of viruses or other pollutant, and then coagulation of them. If you believe the air actually contained the air or originated from biological and chemical, maybe the effect could be worse than could have otherwise. And this is the photo actually looked different, blue sky, where there was a full lockdown and no lockdown. The last one I took myself. And this is also still providing evidence how a human activity linked to air pollution. Rwanda is moving to the electrical mobility. Speaking to the Bogota, which is well known for bus rapid transit, London and Tokyo for subway, and Amsterdam, they have a lot of infrastructure for cycling. And the aim of this actually is try to reduce vehicle emission, a lot of traffic in the city center. So Kigali in Rwanda as well could be also the city where policymakers could look about the reduction of emission using this electrical motorcycle and electrical cars. So, and then this has been evidenced with the research we done last year, where we found if all motorcycle became 100% in the city, could prevent around 70 kilotons of carbon dioxide emission, saved a lot of lives and saved a lot of money where they use for the fuel importation, which means any policy measures also have some positive impact. Very recently, I think the Ministry of Infrastructure launched this automated transit network, where this vehicle may be from Kanombe to downtown, they can use 1 hour. However, using this automated transit, in that case, it could be so quickly in a minute or a few minutes. In that case, you will not need to drive through the city, and this will reduce the traffic across the city center. And I believe this could not only have the impact on the well-being, but will also reduce some air pollution, climate change as well, the greenhouse gases. All these are technology, but as well other solutions, you have to look about the greening space. If you look on the map of Kigali, if as well you are familiar, you will find only in the Kiyovu area, if even you go outside, you may find that we have at least more greening space, more plants in Kimihurura around the Prime Minister office, in Kigali Central Park, in Rugando. So that's where area you can at least we have a very greening area, and the other places to me look empty, and then we still need greening space because they have quite important. There is oxygen, absorb this pollutant, intercept of this particulate, which I was talking, which is the health concern, reduce energy consumption with the trees, and then transpire water and give shade to reduce the air temperature. And more green space, reduce sedentary behavior, increase physical activity, increase mental health, reduce aggression, improve memory, attention, reduce blood pressure. And those are research. And recently in Canada, they found that if you live longer you live longer if you are closer to the green space. So concluding, getting to the end, we still need this technology to reduce this emission, but we need also this more greening space, as well as change our attitude. We have to invest in the research, collaborate, but some requires well a lot of funds. So what next? I recently received, actually yesterday, the funding from the Royal Society, which join other funds from the TWAS and as well as from Canada. And there's a 3 funding we'll be using to invest this chemical and biological air pollution in the school, because the children are more affected at the school. So trying to look how they can reduce their overall exposure. So, and then if you look at monitoring outdoor and indoor in the classroom at Kigali Parent, and this is down outdoor, I correct this air sometime on the filter, and then I will do further analysis on the chemical and biological, looking how this pollutant could affect the school children, as well as have to analyze in a classroom because of different ventilation, air pollution from outdoor may affect indoor or vice versa. So you have to look the correlation between indoor, and this is likely to save the children because we come up with a strong recommendation policy, maybe how will be the classroom infrastructure, or we need maybe what greening space we need around the school, how can we reduce all the traffic emission around the schools. And it's almost important to connect these kids with the nature so that they know that they can do something because they are really mostly affected. So, and then it starts with you, get involved. Thank you.
Wow, thank you very much. Thank you, Dr. Karisa, for this wonderful presentation. I think the participants in this seminar enjoyed it too much. So, dear participants, you are welcome with questions, with request of explanations. So far, there are some questions in the chat room. There is the first one from, from Mukaga Sangwa. She's asking, is there any method used to know where air pollutants are coming from and how to control them? And then Musi Placid is saying, hi, Dr. Karisa. Have you ever tried to assess PM 2.5 and PM 10 and PAH in the areas of industrial zones or power plants in Rwanda? If yes, what is the status in comparison to what you already have? Then another one from Christian Mushimimana. It is a request, I think. Good afternoon, Dr. Karisa. May you share us that PowerPoint presentation you have been using? Thank you. Maybe Dr. Karisa, you can provide these two, the answers for two questions and the feedback for the third request. And if there is any other person wishing to ask a question to Dr. Karisa, she's welcome to use the microphone and ask the question. But maybe before we receive some questions, it's better you provide the feedback on those two questions and one intervention. You are welcome.
Oh, yeah, for the first question is quite easy. So how do we identify the source? So source can be source of air pollution, actually could be identified using also pollutant type. So we know already there are some pollutant which actually originated from different sources. Let's say if we are monitoring vehicle emission, immediately the nitrogen dioxide and particulate matter are those pollutants actually emitted from vehicle emission, where the carbon maybe monoxide and also could come up with different source. So we can know the source either using the tracers, you know, the tracers components which come from different sources. However, we can also use the natural experiment. Let's say if I use the car-free day of COVID-19. So if there is no vehicle in the city, the air pollution is reduced around the 20%, but the next day, once there's the full traffic in the city, then increase around 8%. In that case, you don't need further analysis, and then you can easily tell that the vehicle emission is a major contributor. So there's also quite a lot of modeling we have to do. That's why we sample in different land use so that we can able to know what is the major sources of this pollution. So the second question, have you ever measured this type of pollutant industrial area? Yes, so there's a time I tried to do an analysis actually at Kigali car-free zone, a special economic zone. However, there was no any effect, and this makes sense because we don't have major industries in Rwanda, in Kigali as well. So the industries, the special economic zone, to me, all look like warehouse. They don't have any polluting industry. Even when you visit there, it looks like it's like residential area. So you don't have certain industry were able to contribute to this pollutant, unless you want to go and do specific industrial testing such as Simerwa or iron manufacturing in Rwamagana, but otherwise for the special economic zone we are having now, it's mostly warehouse because I never seen any polluting industry. Yes, so sharing, yes, we can share as well as I think the presentation is recorded as well. Maybe they will share the link. That's fine.
Good, thank you for the feedback. I have 3 hands up. There is the one from Antoine Nsabimana, Julius Nziza, and there is another one who I don't see the name. It is named 2. So let us start by Antoine Nsabimana. You are welcome with your question, please. Come on.
Thank you, Venus, and thank you, Egid, for the nice presentation. I really enjoyed it. So I was wondering if there is a lifespan of the particulated matters. So for instance, I was thinking, how long can you detect, for instance, I remember we have those equipment at the Karisimbi block and the other Ministry of Environment. So the distance, so that you can see the difference between those particulated matters. And if you said that there is no correlation between particulated matters and the microbial diversity or fungus, so how comes, for instance, that with this COVID-19, they are giving us the distance of 2 meters. And for instance, for us, how long can you take those pollutant from the distance between those equipment to take those samples? And now you just said that there is no industries here in Kigali, and by the end, you say, in the conclusion, you said that the pollutant is higher in Rwanda. So where those pollutants are coming from? Thank you.
Okay. Yeah, speaking to the last, so the pollutant, they are coming from different sources, not from from industrial emission. As I said, if you look in Rwanda, most of the vehicles even we imported, you may find they are second hand and older more than 10 years. And those actually, they have been made with traditional technology, which actually likely to contribute more. As well as we have this quite of poor maintenance of these vehicles, which actually likely also to contribute to pollution. And speaking to that as well, our topography of Rwanda make the city or the country a bit to be affected by pollution. Being a country of high hills, any pollution actually emitted, they don't disperse immediately away from the city. However, because of the hills, they make what we call the urban invasion. They become back to the ground. So the topography, the climate, we don't have enough infrastructure. Let's say a car which is moving slowly pollutes more than a car moving faster. So if from Kanombe to Kigali, actually you are in the traffic and you can use 1 hour, and if could be a highway which will use 5 minutes, in that case, it will reduce this type of emission. And this is PM2.5, this particulate, they are not a single pollutant. It's a mixture of different pollutants. So pollution may also come from construction activity, dust emission, cooking using charcoal, because of the emission as well, even though you're cooking inside, depending on our poor ventilation of our house, also they contribute to the outdoor pollution and vice versa. Yes, you may think, I don't say totally the industry could not contribute, but our industry, actually, they have a quite less significant contribution to air pollution compared to vehicle and other human activities. So that's the one. So the lifespan of this, actually this depending on meteorology, how the pollution actually removed from the atmosphere can move through the dry deposition or wet deposition when it rains. So it means after the rain, actually, this emission reduce. Remember, there is this saying in French, "Après la pluie, le bon temps." So it means as well, you may find if we are in the rain season, you may find even the air quality is pretty much clean across the city, because even any pollution emitted is actually removed in atmosphere by wet deposition. So, however, it can also last several weeks while in the air. So to get this, you have to do a long-term monitoring study. So monitoring a week, monitoring a month, or 3 months, 4 months, doesn't give the real picture, especially when you want to set the policy or measure. So you have to set a very long-term monitoring, like more than 5 years, so that you can see a real picture of spatial and seasonal variation of this pollutant. And also there's quite a requirement when you monitor. So depending the objective of monitoring, if you for health purpose, you have to set your monitors for 1.5 to 2 meters, which is the breathing height of the person. But if you want actually to see the source, you can put your monitor on the rooftop on 20 meters so that you can monitor when the air aerosolized. So yes, there was no correlation between chemical and the Chow-1 and Shannon index, which is a species diversity and species richness. However, a stronger correlation was observed between this pollutant with the community, biological community species. Such say some of the species, they strongly correlated with this pollutant. So did I miss anything?
I was asking, for instance, for this COVID-19, they are asking us the distance between 2 meters. And what happened with those ones that you found, the distance? We have some equipment at the Karisimbi and another one to Ministry of Environment. So the distance, is it, did we get any difference significant of composition or some things like that?
Well, speaking to, let's say, COVID, I'll not talk more of this because I'm not sure how this social distancing come. All I know is the air, when they're coming from the source, it's aerosolized in the mixture and mix. So you may find even in the pollution emitted from Rwamagana is actually concentrated here in Kigali city, depending of the wind direction, as well as the wind speed. So in terms of COVID, which the research is still doing, if it's airborne, I can say, so that it can move through the air, and which the research I think is not yet confirmed. But when you said moving the monitors, one in Musanze, another Kigali, yes, that's why I found different land use. This community or chemical, they are different according to the land use. So the pollutant composition of chemical and biological, let's say in Nyabugogo, is totally different of the composition, because I didn't go much showing all the species. But let's say Famikit, which is one of the phyla actually found, was dominant in the rural area. And those Famikit, it's well known as a plant-associated bacteria. And then my sampling was in typical rural location, which confirm why we have a lot of Famikits in the Musanze compared in the rural area. If you look, compared to the urban side. So urban side was dominated by proteobacteria and in PM2.5. So Proteobacteria and PM2.5, those are emitted from biomass. You may find those kind of smoke being in small size, and they are all available in the city compared in the rural location. That's why even the mostly dominated the small size in the city, while the big size actually were dominated in the rural areas. So different land use, they have a higher significant variation, and then they also can even differ from the season, and then this is what I observed. Some pollutant actually emitted from biomass burning. Let's say if you look at the composition of this polycyclic aromatic hydrocarbon, one can give you an example, which is called naphthalene, which is well known from emitted from wood, was dominant in Musanze compared at CST. However, other pollutants such as nitropyrene, which is well known to be emitted from diesel emission, were significantly high at the roadside compared to the Musanze area. And this gives a clear picture of this variation between the land use.
Good, thank you for the feedback. That is related to another question in the chat room. It is from Stash Eustrate Uzawaho. How do you explain Musanze's high pollution levels, which exceed WHO limits despite the fact that it is a rural area? Do you believe it's because of the sinking site, which is caused by the surrounding hills trapping the long distance transport emissions? What about volcanic eruptions' physical properties? What about the high population density? It is a series of questions, but you can look at the chat room. Yeah, come on, please.
Okay, so this is really clear. And then you bring about the nice thing, population by density. And actually why the pollution keep increasing. And this is because we have the highest population per density. I think now you're number one in Africa with the highest population, number 1 or 2 with the highest population in Africa. And this was actually the same case as in New Delhi in India. So despite measures, despite having this highest population per density could be an issue. So although the air pollution in Musanze were actually exceed the World Health Organization, which is true, but it's still less compared to Kigali. If actually pollutant, the concentration of this pollutant in the Kigali is 2 times higher compared to those observed in Musanze. And then as I told you, although being in a typical location, but also is influenced by different other activities such as biomass burning from agriculture, a lot of smoke from wood burning. So all those were the major contributor to air pollution. And as well, there's also slight contribution from pollution in long range transport from the neighboring countries also, which could also contribute. But however, those agriculture, burnings, cookings, they all could also increase the level of this pollutant. So why did you choose, I see another question in the chat. Why did you choose only those pollutants among many other available? Yeah, as I said, there's quite a lot of pollutant, NOx, SOx, carbon dioxide, and so on. I chose this because those are the pollutants of the greatest health concern. So PM2.5, it's actually very toxic compared to other pollutants. And actually, their level has been significantly higher in almost all the countries, and then compared to this pollutant. And then evaluation of air pollution is actually measured only using this particulate. So that was worthy choosing this particulate matter compared to the other because they have less effect. And for chemical composition, we choose this polycyclic because they have been proved to be the first carcinogenic to human compared to the others. So in any way, those are the pollutants of the greatest health concern, which require attentions first compared to the others.
Good, thank you for the feedback. There is also a hand raised up from Julius Nziza. Julius, you are welcome to ask your question, please.
Thank you so much. Thank you, Egid, for a very good presentation. My questions have been answered, but I had another one, actually two. One is, have you, out of curiosity, also compared urban Musanze and peri-urban Musanze areas, especially towards the Volcanoes National Park, because of the conservation and health interests of the country? The second one is, how are you collaborating with the Ministry of Health, particularly in this project, especially to see if there's any correlation of the etiological aspects of respiratory diseases, especially in dry seasons, of the cases that are being reported, especially in these urban areas where you're seeing this, the bacteria and the fungi increased. Thank you so much.
Yeah, thanks, Julius. So the first question, so compare the urban Musanze with peri-urban. Yes, I say not. So because my curious, even in Musanze, I went in the Mukungwa area. It's actually several kilometers from the city center, because my urban areas, I choose the Kigali. So, and then compared to the typical rural location, which is far away from Kigali. So I never done any research in the Musanze city center, so that we can see if there's the influence of volcanic. I don't think we don't have active volcano around Musanze. However, positioning to Nyiragongo as well could also contribute to the both areas, the Musanze and Rubavu. And this is, I remember the first rain after Nyiragongo eruption, I was there. And you find the rains was really dark. It means there was quite contribution of smoke, which was somehow acidic rain, which likely to contribute. But you guys, student here, you are welcome to do your research in Musanze. But we have a station which is far away from Musanze as well, in Mugogo, which is in typical rural area as well. So I never seen any data from Musanze city center, which is actually the city is increasing a lot of rapid urbanization. So which will be worth it to do also research in the urban area. How we collaborating with the Ministry of Health? So far not, and this is really important, because the Ministry of Health should also be part of most of the research, especially for respiratory diseases, because even if you go to any hospital, you'll find the data for hospital admission, mostly those from respiratory diseases are really higher compared to other diseases. However, we are collaborating with the Colorado State University in the US, where we are actually doing a research in Kayonza for around 4 years, where we provide gas and solar panel to some household, and then other we don't provide any. They just continue using wood burning and traditional petrol, something like that for lighting. So, and then we followed them. So where we took the blood pressure, the lung functions, and then after 4 years, we looked the health co-benefit of people cooking using charcoal and wood compared to those cooking using electricity or gas and also solar panel. And this is very likely to provide a very good data, especially health data, because we took the health data for children and the mother who spend quite a lot of time in the kitchen. And this is require a lot of expertise in epidemiologists, which I'm not an epidemiologist now, but I have registered to undertake 3 years in epidemiology. Hopefully in next future, I'll be starting doing all the research, try to link this epidemiological data with the chemical But yes, the ministry should also have the department actually. I think we have the public health, which should be doing also some research on air quality and the health perspective.
Yeah.
Good,
Yeah,
thank you very much. Julius, do you have any other question?
Yeah, just a comment. Yeah, thank you. I think possibly you can also take it from here. It's good to know Egide, and I think for more collaboration, because also I think the University of Rwanda has the School of Public Health. People would be interested in epidemiological studies, and also on my side also the respiratory diseases in the mountain gorillas. So that's why I was asking, especially any data you could be having from peri-urban Musanze area. We can take it after the meeting. Thank you so much.
Good, thank you very much. Do you have to provide the feedback?
No, I think all answered.
Okay, thank you all. Thank you, Carissa, for a good presentation, and thank you for participants. The good questions, interventions that were very helpful for the presenter. If any of you has a research that he wants to present, you are welcome. The center is welcoming all researchers. Every week on Wednesday at 3 p.m. we have the research seminar. So you are welcome anytime for the presentation. Otherwise, I thank everyone who participated and attended this seminar. Let us meet next Wednesday at 3 p.m. Bye, and thank you, Egide, for the presentation.
Thank you. Thank you, everyone, to attend. See you next time. Bye.
Bye.